Gene editing has reached the microbiota. Scientists at Eligo Bioscience have managed for the first time to effectively edit the microbiota in the gut of a mouse.
As you probably already know, the microbiota is the collection of bacteria that inhabit our bodies, from our skin and mucosa to our gut and respiratory tract.
These bacteria have a kind of symbiotic relationship with us. They get nutrients from our body, and in turn, they protect us from infection, help us digest food, and do many other functions.
More importantly, alterations in the microbiota can lead to diseases. Some bacteria proteins can trigger autoimmune or neurodegenerative diseases, and bacteria toxins can cause metabolic and inflammatory diseases or even cancer.
Until now, changing our microbiota was not very easy. While natural methods like editing probiotics can help balance our gut microbiota, modifying the bacteria already in our gut was so far impossible.
In the last years, many research teams have tried to use CRISPR to edit the genes of the bacteria contained within the microbiota, but their efforts have not been very successful.
That was because the vectors they used based on phage viruses were not very efficient in getting CRISPR inside these bacteria. And no CRISPR, no party.
Now scientists at Eligo Bioscience have finally found the key to solving this issue. They engineered a new phage delivery system especially tailored to target the membrane receptor in specific bacteria strains.
This process was not simple, and it required some trial and error and quite a lot of testing. But they managed to develop phages that very efficiently deliver DNA packages to the target bacteria.
And of course, if we want to do gene editing, the package is going to contain CRISPR. In this case, the scientists decided to use base editing and introduce either an adenine base editor or a cytosine base editor in the phage.
And amazingly, when they tested their new gene editing system in vitro, they found that almost 100% of the target bacteria were edited, a very promising result.
An important point to consider is that for safety reasons, this phage vector is not able to replicate after infected bacteria, but having 100 or more phages per bacteria was enough to edit almost 100% of the entire bacteria population.
Okay, great. But you might be thinking that an in vitro result doesn't mean anything, right? So what happens in vivo?
That's exactly the same thing they thought. So to make an in vivo model, they used a strong antibiotic to eliminate the gut microbiota in mice and replaced it with an antibiotic-resistant bacteria strain.
Once these invasive bacteria were established in the gut, they orally delivered the phage carrying the base editor, and they achieved up to 90% of editing in the colonizing bacteria.
And they demonstrated this in different genes, different strains, and even different bacteria species, and that included some non-pathogenic bacteria.
One of the coolest parts of this system is its modularity, which makes targeting a different gene or a different bacterial species a piece of cake. You just need to change either the guide RNA or the phage's surface proteins, or both.
This opens a wide array of possibilities for potential application, from eliminating antibiotic-resistant or pathogenic bacteria to delivering transgenes to superpowering our gut microbiota.
Hopefully, new microbiome-targeted therapies based on this technology will be developed in the future. When that happens, we here with the CRISPR will be ready to tell you all about it.